Hello students, today we are going to discuss about the very important topic of your IGCSE chapter of biotechnology. This is a very important topic in which you will discuss how genetic engineering is done. So, what happens? Firstly, why genetic engineering is done? Why genetic engineering is very important and how it is done. So, to cure diseases, and for biological control, and for the production of hormones, and to produce blood clotting protein as well. Genetic engineering has many other several important features. But today we are just going to discuss about this, the procedure, how we do genetic engineering. First of all, you will take a sample of human cells. First, human cells are taken in that test tube, and these human cells are just processed furthermore to extract the genes. Alright, you can see the human cells are here and they are liquidized. So this is how we liquidize the human cells. This is out of your syllabus. Your examiner is not concerned with it. Alright, what you need to do, you just need to remember the whole process. So human cells are liquidized. You just understand that first of all the human cells are extracted. Human cells are extracted in a test tube and they are liquidized. Alright, and then in the second step, what will happen? Chemicals are added to make the DNA precipitate here. The DNA will precipitate here. You can see here, restriction enzymes are also added. You can see this is the human gene, and the restriction enzyme will cut the DNA at several points. You can see these cutted points and the sticky ends here. These are the sticky ends. What are these? These are basically the sticky ends. So to review it, what will we do? We will take a test tube and we will add the human cells here. And what part of the human cell is more important? The nucleus. The nucleus has chromosomes, chromosomes have genes. So chemicals are added to make the DNA precipitate downward, and the above material is removed. Then what will you do? You will add restriction enzymes. These restriction enzymes will cut the DNA at several parts. At several points the DNA has been cut and you can see the sticky ends. These are the sticky ends which are being produced. In this procedure, you need human cells, especially you need the human gene, and secondly you need the plasmid. What will you take? Secondly, you will take a bacterial cell and you will extract the plasmid. Plasmid is circular. Plasmid is an extra chromosomal DNA and it is circular. It is a circular DNA. So what will be the process here? Restriction enzyme also cut the plasmid. It will make a cut here. It will also produce a sticky end. you can see the sticky ends are produced. Sticky ends are unequal. Blunt ends are equal. You can see the unequal ends. One strand is bigger and one is smaller. It is unequal. Next, so there are two enzymes. Decision enzyme will cut the DNA and ligase enzyme we need here now. What will the ligase do? This is the human DNA. This is the human DNA and this is the bacterial DNA. Human DNA and bacterial DNA, they both will join together with the help of a ligase enzyme. Alright, is that clear? So, ligase enzyme just stick the both DNA together. Ligase enzyme join the sticky ends. And decision enzyme produce the sticky ends of human DNA and plasmids, which are very complementary to each other, so that they can join like that. Now, what will be the next step? This plasmid is then placed back to the bacterial cell. The plasmid is then placed back to the bacterial cell. Now, this recombinant DNA, because it has a human gene and a bacterial gene, this DNA goes back to the bacterial cell. This plasmid acts as a vector. It carries the human insulin gene into the bacterium. The plasmid, this plasmid, is a vector. It carries the human gene into the bacterial cell. Now, after that, what will you do? You will just put this bacterial cell into a culture. And this bacteria will grow when you will provide it with all the nutrients. Lastly, these are the fermenters. In these fermenters, oxygen is supplied, alcohol is removed, all the nutrients which are required by the bacteria are also provided, and a very good temperature and pH is maintained so that the bacteria will grow enormously. In this fermenter, we have millions of bacteria now, and we have millions of the recombinant DNA. Now, what will the scientists do? The scientists will just extract the human insulin gene from the plasmid. So why genetic engineering is important? To cure diseases, for biological control, and for the production of hormones, and for the production of the blood clotting protein. In your book, only the diabetes scenario is mentioned. To cure diabetes, insulin is very important, and this insulin is being produced by the genetic engineering. So first of all, what is done? Extraction of genes. You need to extract two genes. Insulin gene and a bacterial plasmid. First of all, you need to remember that insulin gene is extracted from the human gene. From the human genes, the insulin gene is separately extracted, and from the bacteria, plasmid is extracted. Plasmid is a circular DNA. Plasmid is a circular DNA, extra chromosomal DNA, and plasmid is not associated with reproduction. Plasmid is more associated with the production of the resistance. So plasmid is associated with the resistance of antibiotics. So insulin gene is extracted the way I told you, and the bacterial plasmid is extracted with the help of restriction enzyme. Insulin gene is also extracted with the restriction enzyme. The both of the genes are extracted with the help of restriction enzymes. What do the restriction enzymes do? They cut the DNA, they cut the genes, and they produce the sticky ends. Like that. Unequal ends are produced. Unequal ends are produced here. The unequal ends are produced here. And what are the blunt ends? Blunt ends are like that. Restriction enzyme cut the DNA unequally, but somehow blunt ends are also produced. You can see the equal cuts. Sticky ends are unequal in length. Unequal. Now, both of the genes are cutted with the help of the restriction enzyme. What will be the next scenario? Insulin gene and plasmid. Insulin gene and plasmid, they both are mixed together with the help of a DNA ligase. DNA ligase is an enzyme which joins the unequal sticky ends of insulin and plasmid. DNA ligase is an enzyme which joins the insulin gene with the plasmid. Now, what's the next step? This sample which is now produced is a recombinant plasmid. The bacterial plasmid has a human gene as well. This is a human gene, and this is the plasmid. And this is a plasmid. Human gene and the plasmid, they both are just stick together just because of the DNA ligase. And this plasmid is transferred now to a bacteria cell, to a bacteria. Now, that bacteria is then transferred to a bacterial culture. The bacterial colony will grow, and finally, this bacteria is then transferred to the fermenter. Fermenter have all the required growth medium for the bacteria. They do have a accurate pH level, they do have a required temperature, they do have oxygen supply, they do have nutrients, they do have all the sugars which the bacteria need. So, this is how genetic engineering is done, and this is how insulin is being produced with the help of genetic engineering.